Hello everyone, my name is Lara Becerra and I will be presenting how microplastics affect the gut microbiota. This topic has increased. So microplastics are small units of plastics that are usually less than 5 millimeters and come from larger plastics that start to break apart over time. So the most common types of microplastics are found in the image over here. It's PE, PP, PMMA, and PA. Now, these small fragments are usually ingested or inhaled by humans. Now, the contamination sources can be water, air, and food. However, their main is through ingestion through food and liquids. This ingestion and their presence in the gut has been actually confirmed by studies made after it was found in stool samples. The overaccumulation of these microplastics in the human gut can have an effect on the overall health. Now from this image we can see the three divisions mentioned before: exposure through water, food, and inhalation. However, humans drink and eat more microplastics than they do inhale them. Now that we know what microplastics are and what the gut microbiome does, we're going to be focusing on these research questions to guide us to understand how microplastics interact with the gut microbiome and how these affect our good health. The first research question we're going to be focusing on is how microplastics disrupt the gut microbiota. And then we're going to follow introducing the other research topics. So microplastics reduce the amount of beneficial bacteria, lactobacillus and bifidobacterium, that are present on the gut, and therefore also increase the amount of harmful bacteria present. And the increased amount of harmful bacteria is directly linked to an increase in inflammation. Now, microplastics also cause an imbalance in the gut, which is known as dysbiosis, and is also associated with other digestive issues like metabolic disorders and other inflammatory responses. They also act as pathogens, and this is because microplastics contain toxins and antibiotic-resistant bacteria, and therefore increase the amount of infections and its possible risks. Now, these two images that we see are from a study made showing how microplastic exposure alters the composition of the microbiota over time, specifically in these two beneficial bacteria. Now, the microbiota composition on the control remains rather stable throughout these two bacteria. However, once we have a microplastic intervention or exposure, there's a notable decrease as the time goes on. Now, we're going to be focusing on what disruptions happen after we ingest microplastics. Now, after we ingest microplastics, the microbial diversity is reduced. As mentioned before, the harmful bacteria increases, but the beneficial bacteria decreases, and therefore it impacts the stability of the gut. Now, the relationship between the diversity and the metabolism is explained on the paper published in 2022 by Tamargo et al., where it was explained that the lower the diversity, the weaker the digestion and the weaker the metabolism. The paper also goes into detail exploring how there are changes in the gut microbial diversity, and it's demonstrated by the graphs included in this slide, showing how it does change over time. Now, the metabolic disturbance is suggested by the decline of the microbial diversity, as seen on the difference between the control graph as opposed to the exposed graph. Now, on this diagram, we're able to see the shifts in the gut bacteria that are due to this microplastic exposure. So we have part A, which is the control group, and part B, which is the microplastic exposed group. We can see a significant decrease in the amount of bactericide activity found when comparing the control group to the microplastic exposed group. Like here, we have a lot of blue, about 20, a little bit more than 20%, whereas in here, when the more time passes, there's a significant decrease when comparing it to the control group. Now on this figure, we can see how microplastics impacts the bacteria found in the gut and therefore reducing the amount of short-chain fatty acids. Now again, part A is going to be the control group and part B is going to be the microplastic exposed group. Now on the microplastic exposed group, the amount of beneficial bacteria declines, specifically the amount of bacteria that is responsible for producing these short-chain fatty acids. Now, this decrease in production has been shown to be directly linked to metabolic disorders and a weaker barrier function. Now, this image illustrates the degradation of plastics and the impact it has on metabolism. Now, some gut bacteria will form biofilms and secrete enzymes that break down these plastics into even smaller fragments. Now, this breakdown is not necessarily beneficial because microplastics have chemical additives that when they're released into the body, they will interfere with the metabolism. After this assimilation of the plastics into the metabolism, it will be followed by the complete degradation through metabolic pathways like TCA cycle in this example, to H2O, methane, and carbon dioxide. Now for this last topic, we're going to be focusing on whether microplastic exposure does lead to inflammation and immune dysfunction. Now this image shows the pathway of microplastics and how they enter the human gut, therefore impacting the decomposition of this microbiota. Now this effect is caused because immune cells release a pro-inflammatory cytokinase that triggers inflammatory responses. Now, as time passes and this inflammatory response is still continued, it reaches to oxidative stress, which causes a barrier dysfunction and a potential health effect. Now, we're going to be focusing on these three individual steps. We have part A, part B, and part C, which we will be going into detail. Now, part A, as explained at the beginning of this presentation, focuses on how microplastics enter the digestive system and how they travel through the GI tract. Now, once they enter, step B starts. Part B, we can see that microplastics interact with immune cells in the gut lining, which is represented by this membrane. And when they detect these microplastics, the immune system response begins. Then, on step C, you can see how both molecular and cellular pathways are activated by these microplastics. The ROS, which stands for reactive oxygen species, starts to accumulate, and therefore, because it's very reactive, it causes oxidative stress. Then this oxidative stress starts to build up, and it continues to damage the cells even more, impairing the gut function. And this increases the risks of other metabolic diseases. Then on the other side of this chart, we see P53, which is activated by a cellular stress and regulates this damage response, as well as cell apoptosis, which is cellular death. Then as more cells go through cell death, it leads to a weakening of the gut lining, and therefore leads to an increase in the permeability, making it easier for the bacteria that are harmful to enter the cell. Now, overall, as the oxidative stress starts to build up, it continues to damage the cells even more, and therefore impairing the gut function even more, and therefore increasing the risks of developing metabolic diseases. Now, as shown before, microplastic exposure, or PMPs, as seen on the graph here, which stands for plastic extracted microplastics, as well as TBBPA, which is tetrabromobisphenol A. This is a chemical that is an endocrine disruptor. These two both induce oxidative stress and inflammation. Now on this graph, it is shown experimentally how ROS levels in the gut increase after microplastic and TBBPA are exposed. Now, these levels lead to oxidative stress, which directly damages the cells and it disrupts the gut barrier, triggering inflammatory pathways. Now, as marked as this in this red box, we can see how microplastics alone increase the level of ROS. And this is important because an increased oxidative stress drives this inflammation, leading to conditions like the leaky gut syndrome, immune activation, and other dysfunctions of the gut. Now, microplastics and TBPA together lead to a greater increase in the production of ROS. This correlation is going to be marked by the arrow in red. So the higher the concentration of TBPA, the higher the ROS production, which is indicated in this study by Huang et al., indicated by the fluorescence intensity. Now, why is this important regarding microplastics and the gut? Now, the increased microplastic exposure does weaken the gut barrier, driving inflammation even further, leading to conditions mentioned before. Now, in conclusion, microplastics alter the gut microbiota composition by increasing the amount of harmful bacteria and decreasing the amount of beneficial bacteria. Additionally, the metabolic disruptions that happen after we ingest microplastics leads to oxidative stress and the reduction of these short-chain fatty acid productions. Finally, the microplastics trigger gut inflammation and immune dysfunction, which lead to the previously mentioned chronic diseases. Now, thank you for listening to this presentation, and let me know if you have any questions regarding the content that was covered.